Hello, everyone. My name is Pedro Batista. I am a tenure-track investigator at the National Cancer Institute in the US. I would like to welcome everyone to the RNA Collaborative Seminar Series. We are an international effort that includes over 30 RNA centers. And today's talks are hosted by the NCI and the Penn Institute for RNA Innovation. The talks will be shared on YouTube, so we ask you that please don't make your own recordings or capture screenshots. And just a quick reminder before we start to add your questions in the Q&A box instead of the chat. So it is my pleasure to introduce my colleague, Eugene Valkov. Eugene started his career in the UK, where he obtained his PhD from the University of Oxford. After his postdoctoral research, Eugene joined the Max Planck Institute for Development Biology, where he became a project group leader, and he joined the NCI as a Stadtman tenure-track investigator in 2019. Eugene has a long interest in RNA post-transcriptional processing, and today will share with us a story about messenger RNA decay.
All right. Let me just see if I can share. Okay. Okay, thank you, Pedro, for the introduction. I will tell a story that's actually been recently published from my lab. That's a kind of a slightly unusual departure from us into more of a microbiology world, but still relative to mRNA decay. So the hero of this story is a bacterium called Legionella pneumophila, and it's been termed kind of a covert microbial marauder. And the reason is because it's an organism found in natural and artificial water system, and it's an intracellular parasite of protists and human cells. And it survives inside the cells, and you can see here two types of cells: a macrophage and an infected amoeba. 24 hours, and the little sausages in there are the bacteria, all packed in. And it's able to kind of survive in that environment, which is quite a hostile environment for the bacterium, by modifying the host cell pathways and metabolism in its favor, principally by injecting a whole arsenal of proteins called effectors into the host. So we were inspired in this work by work of Alex Hensminger, who is a microbiologist at the University of Toronto, and Alex's lab really studied Legionella for a long time, and they conducted high throughput screens where they look at effectors interacting with other effectors and effectors interacting with host proteins. And they do this by a variety of methods, most recently by used-to hybrid screens in parallel. And what you see here is the result of one of those used-to hybrid screens, which they published recently. And they identified a small effector called PIF, which is interacting with in one of the panels. They included a set of host proteins, and one of those host proteins was actually something that's of interest to us, which is a NOD7 deadenylase enzyme, which is part of the CCF1 complex. And the arrows indicate interactions, and the thickness of the arrows indicates strength of interactions. And here there's a citation if anyone wants to look up. So PIF belongs on this locus called plasticity. It's a plasticity island of effectors, so pi, which is quite variable even between the strains of Legionella pneumophila. So here are 4 strains, and you can see there's quite a bit of variation. And there are a number of effectors there, which are a function of which is known. And the PIF was really the one that was uncharacterized up until that point. And what Alex's lab have done is they really try to study what PIF is interacting with also by biochemical methods. And this is a result, this is a slide adapted from his paper, where they identified by mass spec, essentially by pouring macrophage lysate onto a PIF immobilized on resin. They identified that really it selectively interacts with the components of the CCF1 complex, which is a part of eukaryotic mRNA decay machinery. So CCF1 is a fairly long-standing interest of mine, and it's been studied by a number of other labs, most notably Elena Contis, Elisa Graudem, Laurie Passmores, and many others. And it's a multi-subunit complex that's conserved in all eukaryotes, and it has a number of subunits. Most of them don't have any enzymatic function. But at the center really is the business part of this complex, and it's the so-called nuclease module. So there are two enzymes, and one of the enzymes is NOD6L, is known as CCR4 or twin in the Drosophila person, and it belongs to the EEP domain, which is a magnesium binding, and it has a leucine-rich repeat domain. Yeast, flies, and worms have just one enzyme. Vertebrates have two similar paralogs of each, which share about 75% identity. And the key thing to remember is that this NOD6 enzyme requires the other enzyme in order to be incorporated into the full complex. It's quite a hierarchical assembly. And the other enzyme is a much more ancient enzyme called NOD7 or NOD8 or CAP1 in yeast. And it belongs to the so-called DEDD superfamily, which is typified by ribonuclease D enzymes. And again, yeast, flies, and worms have just one of those. Vertebrates have two highly similar paralog, which again share 75% identity. And this one binds to directly to NOD1, which is the central scaffold of the CCR4 NOD complex. And then the NOD6L docks on top of that. So this is kind of the architecture that I will use for the remainder of the talk, just to kind of again illustrate. So we like this Pac-Man style cartoons. So I will be showing them, but the main architecture is shown here on the left. And when we entered kind of the fray here, Yevgen, who is a postdoc in the lab, I saw the preprint from the engineering lab, and I said, okay, you want to take a look at this? We're always interested in something relating to CCR4 NOD. And one of the first experiments we did actually nearly killed the project entirely because we found by using a pull-down recombinant proteins that PIF only interacts with just the NOD7 enzyme shown here in the middle. Let me just put the laser pointer on, but not the assembled heterodimers. So the NOD6, NOD7 heterodimer of enzymes, there was no interaction. And on the right here is a positive control with the NOD1. And we then proceeded to kind of study this further. Why is it interacting with one enzyme but not the heterodimer? And we used one of our favorite techniques, which is mass photometry, shown here on the left, to characterize the ternary complex of PIF bound to NOD7 and a little bit of NOD1, the M4G domain. And Claire Pizak, who was a postback in the lab, did a lot of the characterization of biophysically of this interaction. It was a low nanomolar affinity interaction, suggesting quite a tight complex. And then we proceeded to study the activity of PIF on this enzyme system. So this is an assay that we use routinely in the lab. It's a very simple assay where we use a fluorescently labeled RNA oligo with a pseudo poly-A tail of just 20 A's. And what you see here on the left is what happens when you challenge the substrate over a time course with just one enzyme, NOD7. You see a degradation all the way to the sort of right to the very end of the poly-A tail. And when you add PIF stoichiometrically on a 1-to-1 ratio, it inhibits this enzyme very, very effectively. And consistent with biochemical pull-down data, we saw no inhibition of the heterodimer. So the heterodimer of the two enzymes was completely unchallenged by PIF. So to try to kind of understand how this all works mechanistically, in collaboration with Susan Lee, who was the chief of Center for Structural Biology here in Frederick, and using our local facilities here, we determined a high-resolution cryo-EM structure of PIF bound to NOD7, shown here in blue. It presented certain challenges. It's quite a small complex still for cryo-EM, but it was a sub-3 angstrom structure. And as well as bound to NOD7, we also determined the structure of PIF bound to the paralog of NOD7, NOD8, which is very, very similar, and I will not discuss it further in this talk. So when we build a model of PIF bound to NOD7 into the cryo-EM volume, what became very interesting, very obvious, is that the C-terminal region of C-terminal tail of PIF, which is quite loosely kind of structured, assembles into a helix, and this helix then projects into a very interesting part of the NOD7 enzyme, which is right towards its active site. And when we look at the structure of human NOD7 and compare it to the yeast ortholog, structure is extremely similar. This is a very, very conserved enzyme. It uses a divalent magnesium site for activity. PIF really exploits this interface of NOD7, with which it interacts with NOD6. First of all, to dock onto the protein, and then to kind of project its C-terminal extension down towards the active site. So it serves, incidentally, to bind in such a way that is actually mutually exclusive with a cognate partner of NOD7 within the CCF or NOD complex. So in the exciting part of this mechanism, the C-terminal lysine on this helix, on this helical extension, not only projects towards the active site, but actually forms electrostatic interactions with the key catalytic aspartate residues, which are involved in the formation of this divalent site, essentially knocking out the second magnesium and thereby essentially disrupting the catalytic site of this enzyme. So we always do our kind of due diligence with this kind of studies as structural biologists. So we look at all the interfaces and try to assess the importance of various interactions. And the reason, of course, we do this is to design very precise mutants so that we can study them, so we can assess what are the important interfaces and interactions involved in activity of this protein. And we really identified, I would say, 3 classes of mutations. In ring in blue here are mutations which are really disrupting the interface with the other enzymes. So these are sort of NOD6 interacting surface and what we call the docking site of PIF. And then we also characterized the mutations located on this helix, which points towards the active site. And then, of course, we looked also at this final residue, this lysine, and to see what the effects of making mutants there is. And I'll start with the lysine. So replacing the lysine with an alanine essentially alleviated the inhibition of deadenylation by PIF. And you can see this here on the left in the gel. Whereas replacing it with a similarly charged arginine largely retained the inhibitory potency. And this was also reflected in the measurements of binding under equilibrium conditions, which again Claire did, where we saw a significant reduction when we replaced the lysine with alanine, and only a little bit of a loss in affinity when with arginine. So it's a significant contributor to the stability of the bacterial protein to the enzyme. On the other interface, it was a little bit more nuanced. Combining all 5 mutations, so-called 5M mutants, completely destroyed the ability of PIF to inhibit NOD7 deadenylase, and also we detected absolutely no binding. And using just the mutations located on this helix in a so-called 2M mutant, I would say it was like a halfway house. So there was probably about 50% loss of activity or inhibitory potency, and also quite a significant reduction in affinity. So kind of armed with these mutations, we then asked the question: is PIF active against the entire kind of ribonuclease D-type enzymes, D type exonucleases? And another postdoc in the lab, Filip Pekovic, actually had purified PARN, which is again a DED type exonuclease, so similar to NOD7. What you can see here in pink is a crystal structure from a few years ago now, and comparison to NOD7, it's an in silico fantasy. It's an AlphaFold 3 prediction with some RNA bound. But you get a sense of kind of how similar they may look. But the striking thing was that PIF had no impact at all on PARN activity. So it proceeded as completely untouched by this bacterial protein. So given all this, what we were interested in, of course, is to examine the activity of PIF in human cells. And for this, we turned to a technique which we increasingly use in the lab, and it's actually rapidly becoming a favorite, an expensive habit, where we measure mRNA poly-A tail lengths directly from in the cellular context using nanopore sequencing. So this is direct RNA sequencing. And what you see here are two violin plots showing distributions of poly-A tail lengths, and numbers below indicate the millions of reads obtained. So it's a fairly low throughput technique, but you get complete mRNA molecules. And as a control, this was work done by David Turner, who's another postdoc in the lab, so it's a real team effort. When David overexpressed just GFP control in human cells, you get a typical modal tail length around 54 A's. When you overexpress a so-called dominant negative mutant of NOT7, this is where the key catalytic aspartate at position 40 is mutated to an alanine, you get a significant lengthening of poly-A tails to around 146. It's a model value. And the interesting thing is when you overexpress PIF in these cells, you really essentially phenocopy the dominant negative phenotype of a complete knockout of the adenylation, which is this D48. So in red, you can see overexpression of PIF. And as a control to this, we then overexpressed this 5M mutant, which we biochemically characterize as not interacting and not inhibiting activity of NOT7. And the tails then dropped back to their typical model value around 55. And of course, what this does is also create a certain dichotomy with the biochemical results, because of course, biochemically, we could not see an inhibition of the adenylation when the CCF was kind of fully assembled, when we had the functional heterodimer. But here in this cellular context, we saw quite potent inhibition of the adenylation. And the answer really was kind of, partial answer, I guess, to us occurred when we examined the copy numbers of some of the subunits of the CCF complex. We assumed, as I think, a few others who are working on this complex, that they're always present in kind of more or less equal levels in the cells. But that's actually not so. So this is quite an old study now and Martin Beck, actually the first author on this mass spec analysis of human cells. I don't remember which cell type it is. But what became very clear is if you compare the subunits, the so-called core subunits of the CCF complex are present at levels of tens of thousands in the cell, whereas NOT6 is really present at much, much lower levels of just a few hundreds. And when Legionella infects human cells and then multiplies and then injects it to effectors, these are typically injected kind of levels, again, of a few hundreds per cell. So this suggests that the PIF could perhaps effectively compete with NOT6 during the assembly phase of the complex, thereby essentially blocking it and leading to this kind of increased mRNA levels by essentially blocking the rate limiting step of mRNA decay. So question then arose, really, why would Legionella do this? Like, what's the advantage to Legionella of inhibiting the adenylation, inhibiting mRNA decay? What does it gain? And David again analyzed two things. So what you see on the left is the cumulative expansion of cells. And you see that whereas vector control in the 5M mutant can proceed to expand normally, the NOT7 dominant negative mutant and overexpressed PIF kind of really slow down. And of course, we checked for viability as well, so that control was present. And David also analyzed the cell cycle progression as well using flow cytometry. And what became kind of fairly clear is that PIF also phenocopied this kind of stalling of the host cells when overexpressed in the S phase of the cell cycle. So this I'm not an expert, definitely in Legionella biology. I kind of had to educate myself rapidly during the course of this project. But we were wondering why manipulating the cell cycle is beneficial for Legionella. Well, in fact, Legionella, there are several advantages to extending particular cell cycle stages, and this line up nicely with what Legionella needs inside the cell. So during the S phase, the host ramps up nucleotide synthesis, DNA repair, and associated metabolic pathways. And that means there's basically more stuff, more nutrients, more nucleotides available to Regeneron for its own needs. The S phase is coupled to cell growth in preparation for division. This includes synthesis of lipids. And Legionella has a kind of a thing called replication vacuole, with which it sits inside, cocooned and protected inside the host cell. And this requires continuous membrane input and trafficking from ER and Golgi. Perhaps the cell in the S phase is already primed for increased membrane production. And finally, the cells in S phase are under control of DNA damage checkpoints. And many pathogens, Legionella included, benefit from keeping the host alive and metabolically active as long as possible. So biasing the cell into maintaining them into S phase can avoid premature apoptosis or differentiation into a non-permissive state. So why would Legionella manipulate the cell cycle post-transcriptionally as opposed to directly, by acting on cell cycle proteins? So it does that. So other effectors do act on the cell cycle proteins. But I think in this case, one thought that occurred to us, and something that we proposed as a kind of a speculative hypothesis, is that Legionella replicates across a range of different hosts, protists, human cells, macrophages, and many intracellular pathogens manipulate the host cell cycle, but these pathways differ widely between species. So this limits the kind of broad applicability of this strategy. Whereas in contrast, the mRNA decay enzyme, NOT7 in particular, is so highly conserved from protists to higher eukaryotes. So perhaps it's a kind of an attractive universal node for it to kind of hijack for controlling and regulating mRNA homeostasis. So by injecting PIF, Legionella directly manipulates host mRNA levels, including genes controlling cell cycle progression, adding kind of an extra layer of control over host metabolism. So with this kind of, I will kind of try to conclude the story. So we identified this small, otherwise not unremarkable bacterial protein as an incredibly specific and actually the most potent inhibitor of mRNA decay that we had seen in the lab. And we think that PIF likely traps CCF1 in this catalytically incompetent state, possibly during the assembly synthesis. And one thing, of course, we're excited about is using PIF as almost like a template for to design novel and effective deadenylase inhibitors for various either therapeutic applications or even as kind of biological tools. And this is a direction where we're kind of beginning to think about in the future. So with this, it remains for me to thank the people involved. So Yevgen Levdansky, so he led the biochemistry and really led the project. Yevgen will be moving on, so he's leaving the lab and joining the IMP in Vienna early next year. Justin Dime and Susan Lee not shown in this picture. So Susan was the head of the Center for Structural Biology. She's now at St. Jude Children's Hospital, and they really helped us with cryo-EM and really catalyzed the structural part of this project. David Turner here was involved in poly sequencing and cellular assays experiments. Claire Pizak was a postbac, a very talented postbac. It performed a lot of the biophysics and interaction analysis. And Philip here helped with the assays and looking for specificity. And I would just say that we're here based at the RNA Biology Lab in Frederick at the NCI. It's a wonderful community of RNA scientists. And next door to us is also a Center for Structural Biology, as well as the entire NIH intramural community. And I will just finalize by saying that looking to recruit team members in 2026. So if you like what you saw and interested to talk further, please do reach out. Thank you.
Thank you, Eugene. I'll read you the questions from the Q&A box. So Ildar Gainetdinov asks, is NOT6 a bottleneck for regulation of deadenylation? If so, is there any regulation of its abundance or activity during development or in different tissues?
Yeah, great question. Short answer is, so NOT6 and NOT7 do have distinct functions. They're both capable of degrading polyadenosine. They have slightly different levels of activity and specificity for RNA substrates. But what the work from Laurie Passmore's lab and Nari Kim's lab and Jeff Kohler's lab also had shown is that NOD6 and CCR4 really responds to the presence of the poly-A binding protein on the poly-A tail, whereas NOD7 or CAF1 tends to kind of take over and complete the degradation of poly-A tail once the kind of poly-A binding protein peeled off. How it is regulated and whatever context, I think that's still very much an open question. There are two paralogs of each. The literature is a little bit variable on that score, so I would say it's a question mark, no doubt.
I'll ask you a question just to give time for anyone else who would like to ask you questions. I don't know if I've asked you this before. Probably I did, but I don't remember. So are all mRNAs, is there a bias for the mRNAs that are affected by the pi-F interaction, or is it just all mRNAs that are substrates of NOD7? I don't know if your data would allow you to get an insight into that question.
Great question. I'm delighted to say reviewers did not ask me this. So this was on a to-do list, but from the nanopore data, we looked specifically at poly-A tail length as a kind of a global transcriptomic readout. And when, nanopore is a great technique, but what it doesn't give you is depth of coverage. So we got top 20% of all transcripts on that. And of course, to really compare, and identify classes, we probably needed to go to much greater depth with Illumina, but we haven't done that. So that's an interesting question, and I think there's many possibilities there for sure. But yeah, we haven't looked into that.
I don't see any more questions in the Q&A box, so I think we can proceed to the next talk. Thank you, Eugene.
Okay. I think it's time for me to take over here. So I'm Jim Hoxie, director of the Penn Institute for RNA, or co-director of the Penn Institute for RNA Innovation that's directed by Drew Wiseman. I want to thank the RNA Collaborative for giving our institute the opportunity to share this presentation with NCI and this truly worldwide audience. So when we saw that Eugene Valkov is going to be presenting, following up his really interesting Nature Communications paper, we immediately thought of work taking place at Penn in the lab of Dr. Sarah Cherry. So Sarah's lab has long focused on mechanisms that underlie host pathogen interactions, and Dr. Valkov's paper has been of great interest to her group. Sarah's in Japan as we speak, but we are fortunate that Rachel Braun, the student in her lab doing this work, can present. Rachel graduated from Ohio State University. At Penn, for her pre-doctoral studies, she joined Sarah's lab in our cell and molecular biology graduate group. Rachel is a recipient of a coveted NIH Virology T32 training grant, and she's expected to graduate very soon, so all caps, very soon, with the work she'll be telling you about now. So Rachel, the floor is yours.
Great. Thank you, Jim. Let's see. All right, awesome. So, hi everyone. Like Jim said, my name is Rachel, and I am a sixth year, actually biochemistry PhD student, not in chem, but the point still stands. And I'm really, really excited to be here today to represent the Penn Institute for RNA Innovation and to really be able to contribute to this interesting discussion on mRNA decay. So with that, I will be telling you all about the work that I have been spearheading here in the Cherry Lab, in which I have identified and characterized how host mRNA deadenylation very selectively targets interferon mRNAs in order to control both antiviral immunity as well as innate memory, particularly in the context of SARS-CoV-2 infection. Now, in the Cherry Lab, we actually don't have a long-standing interest in mRNA decay, and instead, like Jim said, we have a very long-standing interest in really understanding virus-host interactions. And during SARS-CoV-2 infection, the virus initially targets the respiratory epithelium, at which point the host recognizes that invading virus and activates the innate immune system, which really serves as the first line of defense against viral infection. We also know, however, that various non-immune related host genes can also either directly promote or inhibit viral replication. On top of that, host genes can also be involved in regulating different aspects of innate immune signaling, thus controlling viral replication indirectly. Now, understanding this network of host genes with such regulatory capabilities will not only aid in the development of antivirals, but also aid in the understanding of disease pathogenesis as a whole. So, in efforts to identify host-encoded genes capable of altering the SARS-CoV-2 replication cycle, we conducted a high-throughput siRNA screen in the human lung epithelial cell line called CalU3. We begun by pre-treating our CalU3 cells with our library of siRNAs targeting various host-encoded genes, before then infecting them and fixing, staining, and utilizing automated microscopy to acquire images of these infected cells. From here, we can then run statistical analyses to determine a z-score and to actually quantify the impact of each siRNA depletion on infection. And from these screens, we identified a gene called CNOT2, shown by the orange dot, as a gene necessary for SARS-CoV-2 replication. As you can see here, depletion of CNOT2 leads to a decrease in viral replication, a phenotype that really closely matches the loss of TMPRSS2, which is our positive control gene necessary for viral replication, as it encodes one of the key entry factors for SARS-CoV-2 infection. We also confirmed that loss of CNOT2 with siRNAs leads to around a 1,000-fold reduction in SARS-CoV-2 viral RNA levels as measured by qPCR, altogether indicating that CNOT2 promotes SARS-CoV-2 in this cell system. Now, CNOT2 is just one of those non-catalytic subcomponents of the CCR4 NOT complex, which we all know now is the primary deadenylase responsible for chewing off poly-A residues from the 3' tail of an mRNA, thus initiating its degradation. Now, while CCR4 NOT is super important in the turnover of all mRNAs, it is also known to achieve substrate-specific deadenylation driven by various RNA binding proteins. Now, the class of RNA binding proteins known to interact with CCR4 NOT is the TTP family, comprised of TTP and its two paralogs, ZFP36L1 and ZFP36L2, all of which are known to bind mRNAs via AU-rich elements or AREs within their 3' UTR, shuttling them to CCR4 NOT so that they can get deadenylated and destroyed. Now, because of this, all three of these family members have this pretty homologous tandem zinc finger domain that actually binds AREs within the mRNA, in addition to a highly homologous CNOT1 binding domain that's largely responsible for docking onto CCR4 NOT, serving as a molecular link. Because of these highly homologous functional domains, it's widely believed that when these three family members are co-expressed similarly in similar tissue types, they likely have a lot of the same roles and targets. Indeed, a lot of studies have shown that all three of these family members can bind and destabilize mRNAs containing AU-rich elements, many of which are a lot of cytokines and chemokines like TNF or CSF2. Now, since we had identified from high-throughput screens that CNOT2 promotes infection, we were curious to see if this activity was specific to CNOT2 or more generalizable to the rest of this pathway. Upon testing other factors of mRNA decay, I identified two in addition to CNOT2 that shared the same phenotype. The first is CNOT1, that main scaffolding protein in CCR4 NOT, and the second is TTP, that RNA binding protein that shuttles transcripts to CCR4 NOT. Interestingly, however, the two paralogs of TTP did not show the same proviral phenotype. Loss of either ZFP36L1 or ZFP36L2 had no evident impact on infection, despite their similarities to TTP. Now, once we had identified that TTP-driven mRNA decay factors regulate infection, of course, the next question was, well, what is the mechanism behind this activity? If mRNA decay were really significantly targeting viral RNAs, we would expect it to be antiviral. And since we actually observed the opposite, we hypothesized that instead they might be targeting host mRNAs that impact infection. To validate this model, we first determined the impact of loss of each of these factors on the host transcriptome by by conducting short-read RNA sequencing after individually depleting each subcomponent, and this is just in uninfected, unstimulated cells. From these studies, we found that around 75% of the genes that were found to increase in abundance upon loss of these three factors were interferon-stimulated genes, or ISGs, which are really key antiviral effectors in the interferon innate signaling pathway. During this pathway, the host essentially recognizes the presence of an invading viral RNA and activates both type 1 and type 3 interferons, which can then be secreted from the cell such that they can signal both through the infected cell as well as neighboring cells to activate JAK-STAT signaling. And this ultimately induces hundreds and hundreds of interferon-stimulated genes, or ISGs, which actually go on to inhibit viral replication. Now, it is well known that interferon-related genes are typically maintained at really low to even undetectable levels until viral exposure, at which point they are rapidly induced and then eventually cleared down the line along with viral infection. And this tight regulation is really vital to cellular homeostasis because while this pathway is super important in antiviral defense, excessive signaling is also problematic as it can lead to complications like inflammation or autoimmunity. Now, while a lot is known about this induction of interferon, less is known about the precise molecular mechanisms that maintain low basal levels at steady state or that bring them back down to baseline post-stimulation. Given this and our RNA sequencing results, we hypothesize that mRNA decay could contribute to this control. Indeed, I did find that when I deplete CNOT2, CNOT1, or TTP in uninfected Calu-3 cells, we do see increased levels of both interferon beta and interferon lambda-1 mRNA, in addition to two examples of downstream ISGs, IFIT1 and TRIM22. Altogether, this implicates important roles for this machinery in negatively controlling interferon mRNAs at steady state, suggesting that these factors could be responsible for that low basal level. Now, like I said earlier in this talk, these factors of mRNA decay, most notably the TTP family, have really well-established roles in regulating a lot of pro-inflammatory cytokines and chemokines that harbor AREs. Now, naturally, we were curious to see if some of these classical targets were also being regulated in our system. Here I'm just showing two examples as examples. One is IL-8 and the other is CXCL1, both of which have AREs, but surprisingly, neither of which seem to be regulated by this machinery in our system. Depletion of these three factors has no noticeable impact on these transcripts. Now, this has two interesting possible implications. First, being that an ARE alone could actually be insufficient to drive this selectivity. And second, this data suggests that TTP could actually have different types of targets in a cell type specific manner, where in lung epithelial cells like this, it specifically regulates interferon related mRNAs rather than some of these classical pro-inflammatory cytokines and chemokines. Now, we were also interested in looking at this phenomenon through another lens of specificity, and that is whether the paralogs can also accomplish this regulation of interferon. However, we found that depletion of either ZFP36L1 or ZFP36L2 actually does not lead to an increase in either interferon or ISGs like depletion of TTP does. And this was especially intriguing to us because of this highly homologous TNM zinc finger domain. Theoretically, all three of these proteins are they're all expressed in these cells, so theoretically they should all have that ability to bind ARE containing targets like interferon. Now, one possibility that we did consider when it comes to this is the possibility of redundancy. If these paralogs all have similar targets and similar roles and we're only taking out one of them, the others could still be there to compensate and explain why we're not seeing a detectable phenotype. So to account for this possibility, I conducted concurrent triple knockdown of all three paralogs at once, in addition to doing individual knockdown of each. And if we look at two targets that have been shown in previous studies to be regulated by all three paralogs, we do indeed see that they seem to be regulated redundantly. And the TNF and CSF2 are the two targets I'm talking about. While depletion of just one factor might lead to some phenotype in some cases, knockdown of all three leads to the strongest increase in TNF or CSF2 mRNAs. When it comes to interferons, however, this actually is not the case. Instead, knockdown of TTP alone leads to the strongest increase in interferon mRNA levels, suggesting that while some targets like TNF or CSF2 are controlled redundantly by all three family members, for some reason, it seems that the regulation of interferon is highly, highly specific to just TTP. Now, we know from previous work that interferon signaling is incredibly potently antiviral against SARS-CoV-2. Here I'm just showing that pretreatment of cells with either interferon beta or lambda-1 leads to a decrease in SARS-CoV-2 viral replication. Given this, in addition to the fact that we've identified mRNA decay factors that negatively control this pathway, we hypothesize that it is through this negative control that these factors promote infection. So to validate this mechanism, we implemented a drug called ruxolitinib, which is a JAK-STAT inhibitor that blocks downstream production of ISGs. As I showed you guys earlier in the talk, in the absence of this drug, depletion of our factors leads to a decrease in viral RNA. However, when ruxolitinib is added, this phenotype is entirely lost, and these depletions have no impact on infection. This suggests that this proviral activity is indeed totally dependent on the signaling pathway supporting our model. Next, we wanted to pinpoint the exact step in the signaling pathway actually being targeted. To do so, we again implemented ruxolitinib, this time in uninfected cells, to look at levels of interferon mRNA. And importantly, we found that that increase in interferon beta mRNA upon loss of these factors occurs regardless of whether JAK-STAT signaling is intact or not. And this leads us to believe that this regulation is direct to interferon mRNAs themselves, and that this increase that we're seeing is not just some artifact of feedback or lingering signaling. We also wanted to take this one step further to determine if this regulation is driven by the deadenylation activity of these factors. So CCR4 is the primary deadenylase in the cell. Is that deadenylation activity what's causing this regulation? To do so, we implemented nanopore direct RNA sequencing to measure the poly-A tail lengths of interferon mRNAs before and after loss of CNOT2. Here I'm just showing interferon beta as one example, but we did find for many interferon mRNAs that loss of CNOT2 led to a longer poly-A tail length compared to control. This indeed does suggest that these interferon mRNAs are sensitive to targeted specific deadenylation by this particular machinery. Now, it's well studied that shortening the poly-A tail can lead to the degradation and destruction of an mRNA. However, poly-A shortening is also implicated in other methods of gene expression, one example being potential regulation of downstream translation. So to determine if poly-A tail length could impact translation at all in our cell system, we teamed up with Norbert Party to transfect Calu-3 cells directly with interferon beta mRNA harboring either a 101 or 51 nucleotide poly-A tail. As a control transcript or a comparison transcript, we also transfected cells with GFP mRNAs harboring these same two tail lengths. And if we do this and then utilize qPCR to measure the mRNA levels of those transfected in mRNAs, GFP, and interferon beta, we can see that the expression of these mRNAs is more or less the same regardless of tail length. A, suggesting that input is about the same, and B, suggesting that these two mRNAs don't really have any differential stability regardless of these two particular tail lengths. When we look at downstream interferon signaling, however, the results actually look a little bit different. Here we see that cells transfected with the longer-tailed interferon beta actually produce more ISGs than those with the shorter-tailed interferon beta. This suggests that shorter-tailed interferon betas might actually be translated less efficiently than those with a longer tail, implicating that tail length could indeed play roles in determining translation potential. We also confirm this at the protein level by confirming that protein levels of the ISG TRIM22 are produced more in cells transfected with that longer-tailed interferon beta than the shorter-tailed. Now, interestingly, we found that when we look at GFP in cells transfected with these GFP constructs, it seems to actually be translated equally well regardless of tail length. And this is an interesting finding because it suggests that certain mRNAs, like interferon mRNAs, or maybe even others that are also highly turned over, could actually be more sensitive to a shortened poly-A tail as an added mechanism of control. Altogether, this suggests that this targeted deadenylation by this machinery could indeed control interferon mRNAs, at least through one avenue, by regulating their downstream translation potential. Like I said, however, the main mechanism by which poly-A shortening impacts gene expression is through mRNA decay and regulating stability. So to determine if the stability of interferon mRNAs are regulated by this machinery, we utilize this tool called diabsy, which is a small molecule sting agonist. All you really need to know about diabsy for the purpose of my talk is that it very transiently induces interferon, leading to this rapid accumulation of interferons and ISGs followed by an equally rapid decline back to baseline, which I'm showing here for 3 representative ISGs. And we felt that this tool would be useful to determine if the kinetics of these signaling events are at all impacted by loss of CNOT2. And indeed, I found that loss of CNOT2 does indeed lead to a more sustained interferon response, and these ISGs appear to be more stabilized, altogether implicating roles for this machinery and also bringing interferon mRNAs back down to baseline post-stimulation, and suggesting that on top of regulating interferons through translation, they're also destabilizing them through that deacetylation activity. Now, this finding that loss of CNOT2 leads to this really sustained interferon response led us to look at one other mechanism by which interferon signaling is regulated, and that is at the level of cellular priming. Now, as I mentioned earlier, interferon-related genes are typically maintained at very low levels until primary stimulation, at which point they're induced. Previous studies have actually shown that this initial induction can lead to cellular changes that remain persistent even when the cell has transcriptionally recovered, such that when a new challenge is added, like virus or new interferon, the cell is actually poised and ready to respond more quickly to reactivate those ISGs even more quickly than that first stimulation. And overall, this phenomenon is known as priming or innate memory and has been shown at the level of various ISGs. Here I'm just showing one example, but there are many different studies that show that hitting cells with a pulse of interferon, letting them recover, and then introducing a new pulse of interferon will actually lead to an enhanced induction of the ISGs. GBP5 and GBP1 are 2 examples here, compared to cells that did not receive that initial dose. Now, since we had found that loss of CNOT2 leads to this really sustained interferon response, we were curious to see if depleting CNOT2 and perturbing this targeted decay of interferons could prime cells in a way like this. So essentially, the proposed experiment here would be to deplete CNOT2 with our siRNAs, let the cells recover, and then introduce some secondary challenge to see what happens. And our first question is, well, how long does it take cells to recover? And to cut to the chase, this is very exciting. We found that on day 10 post-depletion, CNOT2 levels are back up to baseline and interferon mRNA levels are back down to baseline. So we chose to move forward with a minimum window of 10 days. We were first curious to validate the phenotype that others have shown, right? So does priming lead to enhanced reinduction of ISGs? To do this, we implemented diabsy, that same small molecule interferon inducer, on day 10. And indeed, we found that cells primed by loss of CNOT2 10 days earlier did see a more robust activation of this ISG, IFIT1, compared to control cells. Interestingly, however, we also found that the same phenotype held true, if not to an even greater extent, for the upstream interferons themselves. In the case of both interferon beta and interferon lambda 1, depletion of CNOT2 10 days earlier leads to a much more robust activation of these interferons compared to cells primed with just a dose of SI control. Now, this was especially exciting to us because while previous studies have shown that priming can affect the reinduction of these ISGs, to our knowledge, this is the first time that it has been shown that priming events can also lead to a faster response for interferon mRNAs themselves. Now, since we found that these interferons are so rapidly induced by this new stimulus, we essentially hypothesized that these priming events were leading to some sort of cellular change that is then poisoning the cells such that they can retranscribe interferon more robustly than a naive cell. To test this, we also probed the activation status of the transcription factor that activates interferons, IRF3, whose activation can be monitored by its phosphorylation. So inactive, it's unphosphorylated; active, it's phosphorylated. As you can see here, for cells that don't receive a secondary challenge and instead are just hit with a dose of DMSO, we see no evident phosphorylated IRF3. In the acute condition, this lack of phospho-IRF3 is consistent with our hypothesis that the induction of interferon that we're seeing at this time point is due to a loss of decay rather than new transcription of interferon. In the case of the prime cells, however, this lack of phospho-IRF3 is just showing that the cells at this point are in an off state. There's no feedback or lingering signaling that's causing new induction of interferon transcription at this time. Now, when we add in diabsy as a secondary challenge, while we see accumulation of phospho-IRF3 in all conditions, we see a much stronger accumulation in those cells primed by loss of CNOT2. This indeed does confirm to us or suggest to us that these priming events are leading to some sort of cellular change that remains persistent and changes signal transduction essentially, such that when a new stimulus is added, like this pulsate diapsy, cells primed by loss of CNOT2 are able to more quickly and more robustly activate IRF3 to transcribe those interferons. And this enhanced ability of the cell to reactivate interferons is also consistent with the persistent ability of the cell to still protect against viral infection. So even when CNOT2 is depleted 10 days earlier, we still see that that cell is protected against viral infection, despite the fact that CNOT2 levels have entirely returned back to baseline. Now, just to summarize this memory model, we found that loss of decay factors induces levels of interferon mRNAs because of that loss of decay. This leads to a natural downstream production of ISGs, which we are hypothesizing poisons the cell such that interferon transcription is in an off state, but the cell is on standby so that when we add a new secondary challenge, it is able to more quickly activate IRF3 to transcribe these interferons and be ready to tackle that secondary challenge. Now, importantly, what I want to remind you guys of from the beginning of this presentation is we found that this targeted deacetylation is highly specific to TTP. It's not accomplished by the two paralogs of TTP. And that is really what all of my current and future work is focused on. How? What is the precise molecular mechanism behind this specificity? Now, in these endeavors, we have first identified that TTP's expression and stability are both regulated by viral infection itself. SARS-CoV-2 infection induces levels of TTP, but not the two paralogs. I've also found that TTP is induced by other non-virus interferon inducers. Here I'm just showing this synthetic double-stranded RNA mimic poly-IC as one example. Now, the fact that TTP is induced by the same stimuli that also induce its interferon targets suggests to us that TTP's stability, activity, and availability are all super important to its ability to regulate these targets. Another major goal is to conduct mutational analyses to determine exact functional domains in TTP necessary for this selectivity, which of course, to do this, we first need to establish an overexpression phenotype for the wild type protein. I have again been implementing direct transfection of TTP mRNA for these studies and have indeed found that ectopic expression of TTP leads to lower levels of interferons and ISGs in uninfected cells relative to cells ectopically expressing GFP. This is also consistent in infected cells with higher infection in the cells overexpressing TTP compared to GFP. So now that we've identified this wild type phenotype and confirmed that overexpression of TTP has the opposite impact to knockdown of TTP, we can now hopefully introduce some key functional mutations or maybe even swap domains between the paralogs to try to identify which parts of TTP drive the selectivity. Along similar lines, we are also hoping to conduct similar mutational analyses this time to determine the impact of genetic variants in TTP. There are indeed studies which show that polymorphisms in TTP can be associated with inflammatory conditions. Most notably is the single nucleotide polymorphism at the start of the tandem zinc finger domain, which surprisingly doesn't even change the amino acid sequence of TTP, but instead introduces the rarest arginine codon and has been shown to slow TTP translation, leading to a loss of function. And the SNP is associated with an inflammatory condition known as rheumatoid arthritis. So we were curious to see if polymorphisms like this could also impact patients' susceptibility to disease. And we found that when we ectopically express this RA-associated TTP, we do see lower levels of infection relative to wild type, in addition to a modest increase in interferon mRNAs relative to wild type. And this is all consistent with lower protein levels of TTP, despite the fact that RNA input is the same. All of this is really just a proof of principle to show that polymorphisms in TTP definitely do have potential to impact susceptibility to disease or disease outcomes, and also gives us some hope that tuning TTP's activity or expression might serve as a promising avenue for therapeutic intervention. So to summarize, we have identified this highly specific role for the CCR4 NOT complex and TTP in post-transcriptionally regulating interferon mRNAs to maintain their low basal levels at steady state. Stimulation of the cell not only induces interferons themselves, but also induces TTP itself, making it abundant and available to contribute to this vital return to homeostasis. Because of this induction and because of this negative regulation of interferon, virus is actually able to benefit from this as these factors are indeed proviral. And finally, we have identified a role for this machinery in cellular priming by showing that loss of it or interruption of it prime cells for a more enhanced induction of interferon down the line. So since TTP is really the key driving factor behind this selectivity as the RNA binding protein, and because we just learned from Dr. Valkov's talk that global inhibition of mRNA decay by inhibiting the catalytic domain could have functional consequences or could even benefit a bacteria or a virus in sort of a host pathogens arms race scenario. We are proposing that tuning TTP's expression or activity, since it is very selectively targeting interferon mRNAs, could serve as an interesting avenue to look at therapeutics for things like diverse infectious diseases or maybe even autoimmunity. And with that, I would like to thank everybody in the Cherry Lab, as well as our many collaborators in the high throughput screening core at Penn, the Lynch, Dice, Blackshear, Gerardo, and Pardee labs, in addition to my thesis committee and funding sources. And above all else, I would like to thank the RNA Society, the NCI, and of course, the Penn Institute for RNA Innovation for allowing me to represent the Institute today. And I'm happy to take questions from anybody.
Thanks, Rachel. Floor is open
Yeah.
for discussion. Looking at the chat box, nothing yet. So two talks really rich and getting us to think about the delicate balance between the pathogen and the host. Rachel, can you comment? So I know Sarah thinks about a lot of viruses besides SARS-CoV-2. To what extent do you think these themes play into other coronaviruses? We've got MERS, SARS-1 to think about. Where does the lab think about these other?
Yeah, so I've looked at the role of this machinery in other viruses as well, surprisingly less so in other coronaviruses. We early in my PhD, we started looking at some of the common cold ones like OC43, which actually don't infect these cells, so we stopped those endeavors. But I can say with confidence that these factors also promote other RNA viruses that are sensitive to interferon. Like the two examples I've looked at most are influenza A and vesicular stomatitis virus. Those are less sensitive to interferon than SARS-CoV-2. SARS-CoV-2 is like especially potently inhibited by this pathway. So we see a much stronger phenotype for SARS-CoV-2 infection. But those other interferon sensitive viruses, we do still see a decrease in them if we deplete this machinery just to a lower magnitude.
Okay, so there's a question from Fabrice Leclerc. I'll read it. Approximately 25% of severe SARS-CoV-2 infections are attributed to low interferon levels, Casanova Lab. Could the described mRNA decay mechanism contribute to the proportion of severe cases? If so, which risk factors among the regulators you identified might be implicated? Are you aware of any known gene variants among those regulators that would favor mRNA decay?
Yeah, loaded question. I don't have any evidence. Obviously, we haven't pulled data from patients yet. That is a major future goal, though. But that is kind of what we're hypothesizing and what I am trying to get at when I look at that like rheumatoid arthritis associated SNP is the possibility that genetic variants in this machinery could maybe be found in patients like that have either a deficient or an enhanced interferon response, really, because while it's often, and I think more often shown that patients deficient in interferon signaling or with, dysregulated interferon signaling have worse infection, it can also happen the other way around. Patients that are producing too much interferon can also have poor disease outcomes. So from what I know of, at least between TTP, C01 and C02, the only one I've been able to find patient information on genetic variants for is TTP. And since that seems to be the selectivity factor, that's probably where I would look first to see if there are any patient-associated polymorphisms within TTP in patients that have, those more severe ends of infection outcomes. But I feel like there was a last part of that question that I forgot about.
Yeah, so we have 1 minute and one more question from
Okay.
from, from Nils Walter. Rachel, great talk. Can you review how exactly the memory associated with the priming is stored, given that any protein factors or signaling pathways either get degraded or have countermeasures reducing them?
Yep. We're still working on fine-tuning and refining that mechanism, but we have some idea. A lot of the time, like traditionally, memory is found to happen at the level of epigenetics. So priming events, whether it be by interferon or a virus or something, often lead to different epigenetic changes, whether it be chromatin marks or something like that. And that can lead to a memory or priming response like this. We did some very preliminary attack-seq studies to see if something like that was happening in our system. And we didn't see any super noticeable changes in chromatin accessibility at the loci. And instead, that led us to look at signal transduction. And essentially, it's actually, I think, a pretty simple model. What we think is going on, it seems to be more of a feedback loop. So induction of interferon and ISGs can lead to that accumulation of a lot of ISGs, which actually feed back into the system to reactivate interferon. So like IRF7 is a common one that is an ISG and it's induced by this pathway, and then it can feed back to induce the interferons themselves even more. And what we think is happening based on our data so far is essentially that induction we see when we deplete CNOT2 is leading to an accumulation of very upstream components so that they're accumulated and they're more abundant than a naive cell, but they're actually not firing. So they're not causing new interferon transcription. They're just more abundant and poised so that when the new challenge comes in, they activate much stronger than like a naive cell that doesn't have those. So that's what it seems like is happening so far.
All right, lots to do looking ahead, and I think we are at the hour. I see there are no more open questions, so
Awesome.
unless we have comments from our host,